Hello, today we have the pleasure of speaking with David Furman, PhD from the Furman Laboratories. He's an associate professor at the Buck Institute, and he'll be discussing the role of longevity as it applies to space research. And David will be one of the speakers at the upcoming HEALinc event happening in the Bahamas on April 17th. So David, thank you very much. So tell us what you're doing at the Buck Institute and the Furman Lab.
Right. Thank you so much for having me. The basis of what we're doing is to use the immune system and understand how we age in different organs and come up with interventions that will normalize deviations that some of us will suffer from. And those deviations in the immune system are largely defining how we age. In the cardiovascular system, in the neurodegenerative disorders, musculoskeletal conditions, etc. And that's overall what we do. Around, I would say 3 years ago, we started working with NASA. NASA reached out to us for our expertise in inflammation and aging. And they mentioned that astronauts are suffering from a form of accelerated aging. That opened really a number of opportunities within NASA and outside of NASA with private companies that are sending rockets into space. And so it's really interesting what happens because when a human will go in space, during that time that they spend in space due to microgravity, they accelerate a number of different biological aging processes and networks. And that's what we're using now to understand accelerated aging and be able to develop interventions to tackle aging-related diseases and extending lifespan and health span.
Well, it's certainly a very interesting physiological model that you've stumbled upon. Obviously, it's not something you would correlate for space travel, but with space stations now being deployed around the world and a lot of the international collaboration, I'm sure it's a real issue that people are looking at in a very interesting test tube, if you will, to observe some of the phenomena of aging. What are some of the tools that you're using to measure some of this or to validate or quantify it?
So it's interesting that you mentioned the test tube because we actually have now simulation of microgravity and galactic rays on the ground. So we are able to use a system that was developed by NASA, and we have a facility of microgravity simulation at the Buck Institute where we take human cells, but also human organoids. These are mini organs about half of a millimeter in size or diameter, and these are absolutely functional organs that are so small that we can put them into a system that accelerates their aging process by simulating microgravity. And so that is one of the tools that we're using. And we're measuring hundreds of thousands of compounds in these organoid systems. We do transcriptomics, we do proteomics, we do metabolomics, all the possible omics that you can think of to really understand what are the pathways that get accelerated during microgravity. And then we overlap those with data from astronauts that have flown in space. In addition to that, we have a program that enables us to go from this multi-omics assessment of organoids, human organoids that is, to interventions. And so you can imagine that because of our high throughput system in which we can test many, many interventions in a short period of time, in 24 hours, approximately 25 hours, we can accelerate the aging of those organoids by 50%. This means that using this system, we can really test a number of different interventions to reverse those phenotypes of aging and bring them back to an optimal state.
What's really interesting about what you're doing is you're actually combining several of the most novel and modernly advanced developments in life science technology. So you mentioned organoids. And back in the day, we had the animal models, we've had chimpanzees, we've had all these systems that we've tried to model the effects. And now literally you can create organ-specific or species-specific specimens that you can try all the cause and effect relationship experiments on. And I think it's interesting that you're looking at all the different omics. You look at proteomics, metabolomics, genomics. So we're looking at the influence of the physical parameters, right? So we have gravity and radiation, I guess, are the two you identified, and how those affect fundamental biology of humans and how we express our proteins and how we metabolize things that we need for energy. I'm sure there's a lot more to be done, especially as you translate to the immune system and the mitochondria and looking at those interrelationships as well.
You're absolutely right. So there are a number of different biological pathways that will be discovered and are being discovered at the moment when you integrate multi-omics platforms and you use AI and machine learning to make sense out of the data. And then so one is to discover whole networks of pathways of aging that change during microgravity. Then a very different thing is to go from that biomarker discovery platform to interventions. And that's what we're doing here at the Buck Institute. Also, a startup company that we formed not long ago, Cosmica. We're going to be presenting that at the symposium, at the summit, alongside with a new consortium that we have started to really advance the field of space medicine and longevity.
I think it's amazing the research you're doing and how it can actually translate to something tangible. One of the problems with life science research is it's a long series of processes before it actually can get to a patient and make a difference. This looks like it's kind of on a fast track where you're looking at processes that can have a direct effect in clinical practice sooner than later.
Yeah, it's interesting. A new way of looking at the biology of aging. It really bypasses all the model organisms. I'm not sure if you're familiar with the drug discovery timeline, but it typically takes about 10 to 15 years to develop a drug and deploy that into the marketplace, and about $1.5 billion. And the reason is we're looking at the wrong models. Mice are not the same as humans. 65 million years of evolution will separate mice from humans, and not to mention worms or flies. So we're focusing on human biology. Chances are translation into human biology will be higher. We'll have a higher success rate, and we could do this at an accelerated fashion. Just as an example of this, we're 12 to 16 months out to initiate our first clinical trial, and we started this a year ago. So we're maximizing the effort and every dollar into developing these interventions for aging that are much more improved as compared to what you can get from animals. And two, the chances are that the translation into human biology will be higher. So it's really interesting. The other thing that I would like to mention is when you have this accelerated model for aging, an organoid system or a cell is put back into 1G, and this is a normal gravity, there is a rejuvenation process. So when I mentioned astronauts suffer from accelerated aging at around 30-fold in every single organ, that doesn't mean they will die in 2 years. They come back to Earth and they actually recover. For the most part of their biology will recover. So this means that there are geroprotective, naturally occurring mechanisms that we're now being able to quantify and measure, and we can develop mimetics of those compounds to really develop sets of new classes of geroprotectors.
That's incredible. And I think that the meeting that Desiree put together, I know that's one of her goals, is to take whatever collaborations and efforts we have from this meeting and expand them to people that have worldwide aspirations. And I'd imagine that what you're doing is really going to have lasting implications for many aspects of longevity. So we look forward to seeing you there. Is there anything else you'd like us to know about the research you're doing or what we're planning to do in Atlantis and the Bahamas?
I'm very excited to get to meet folks there and to share our vision to really impact humanity in a positive way in the long term. That's all I wanted to mention. Thank you so much for having me.
Yeah, thank you, David. Well, congratulations on all the great work you're doing. And we look forward to seeing you in just a few weeks in the Bahamas.